Hello everyone, welcome to Get My Class. So let's solve this question here. It says arrange the following in the order for Hershey Chase experiment. So it should be in the order of Hershey and Chase experiment. The first one says radioactive phages were allowed to attach to E. coli cells. Yes. The second one says the virus particles were separated from bacteria by spinning them in a centrifuge. The third one says as the infection proceeds, the viral coats from the bacteria were removed by agitating them in a blender. Okay, they have given various steps, but we have to arrange them in an order. That's what it says. Arrange the following in an order of what? Of Hershey and Chase experiment. And here they have given various orders as the options, that is A, B, C, D. So before we look into the correct option, first of all, we have to understand what is this Hershey and Chase experiment. We all know about Hershey and Chase experiment, where they performed an experiment for what? To look for the DNA, or you can say to check the nature of the genetic material. Okay, to check for the nature of the genetic material. Okay, so they wanted to look for the nature of genetic material because Griffith, when he performed, he said that the genetic and material is necessary for the transmission of the characteristics, but he did not know what is the nature of this genetic material. But because of Hershey and Chase experiment, they came to know that it is the DNA, the genetic material is DNA, and DNA is responsible, or DNA is the transforming principle. Transforming principle. Okay. So what did they do? They first, okay, they treated the phage. Okay, they utilized phage, bacteriophage, which actually brings about infection of the bacteria here. So what did they do? They took phage, and in one set, they cultured phage in the medium containing radioactive sulfur, radioactive sulfur, and then they allowed it to infect the bacteria. Okay. After infection, they allowed it to proceed, prolong the infection, and then further, they wanted to separate the phage particles from the bacterial cells because they would have bound tightly to the cells, bacterial cells. So for separation, what did they do? They actually they did you can say they blend this mixture, or by you can say they agitated it actually. Okay, they agitated means they blend. They put the mixture in the blender so as to separate the phage particles from the bacteria. And further, they performed centrifugation. What did they do? They performed centrifugation. Okay, when they did centrifugation, for their surprise, they saw that the bacterial cells, they form the pellet, of course, because they are heavy. What is this? This is E. coli cells. These are E. coli cells. Whereas on top is the phage particles, they formed a band, and these were radioactive. Why? Because these were radioactive because they contain the radioactive sulfur. Because the component, the major component of the phage is nothing but the protein. Okay, so they were radioactive, whereas the pellet, that is the E. coli cells, were not radioactive. Okay, so then what did they do? They took another set where the phage particles were now grown on or grown in the medium containing radioactive phosphorus. They used radioactive phosphorus. Now they saw that the phage particle, of course, the genetic material is what? DNA, and phosphorus is very important for the formation of the DNA, right? And hence they saw that, or they observed that there was incorporation of the phosphorus in that particular DNA of the phage. So that means the DNA is radioactive now. So when these phage particles were allowed to infect bacteria, okay, and further, what did they do? They, of course, they agitated, and then centrifugation. Centrifugation. Okay. Here they saw that in the test tube, this is the pellet and this is the band of phage. The band of the phage was not radioactive. In fact, the E. coli cells were radioactive. E. coli cells. These were what? These were radioactive. And how is that possible? Because they did not culture bacteria in the radioactive material or medium. How did they become radioactive? That means the phage has allowed or incorporated its DNA, which was actually radioactive, in the initial stage itself. And that is how the E. coli cells are glowing now, or they are becoming radioactive, right? Okay, so that's the experiment. And here it shows that the DNA got incorporated into the E. coli cells, and hence DNA is the genetic material. That is how they proved. Okay, so let us see. So the basic steps what they have mentioned here, radioactive phages were allowed to attach E. coli bacteria. That is the first step. That is absolutely right. Okay, this is the first step. The second step, they say that the virus particles were separated from bacteria by spinning them in the centrifugation. No, this is the last step. So this third one says as the infection proceeds. Yes, so first they allow the bacteria phage to infect. So as the infection proceeds, so this should be the second step. Okay, then the viral coats from the bacteria were removed by agitating them in the blender. Yeah, this is the third step. And hence after that, after blending, it will be subjected to centrifugation. That will be your fourth step. That is the second one. So it should be what it should be. It should be 1. The first point is 1. The second point is 3. The third point is 4. And the fourth the fourth step is the second one according to our explanation. That is 1, 3, 4, 2. Let us see. Option A says 1, 2, 3, 4, which is incorrect according to our explanation. Option B says 1, 3, 4, 2. This is absolutely right because we also got the same step. 1, 3, 4, 2. Option C says 4, 3, 1, 2, which is incorrect. D says 1, 3, 2, 4, which is again incorrect. So what is the correct option? The correct option is option B. I hope you understood the explanation. Thank you.